Advika, just give me few minutes, okay?
Okay, ma'am.
Okay, now we can start. Advika, what about yesterday's homework?
Ma'am, I wrote my notes. The bacteria drawing I have to do
When you're going to do that?
after class. Like that is the only thing left.
Okay. Till where we have completed? I think we have completed. We have discussed about the cell number. We have discussed about the prokaryotic, eukaryotic, right? Okay. Now we will be discussing about the uh, ultra structure of the cell. Or I mean like eukaryotic cell, what are the structure they will be having?
Yeah.
Is it visible?
Yes, ma'am.
Okay. Hmm. Now here, the first part what we will be discussing is the cell wall. Now cell wall is present whom? In the plant or in the animal cell or in both?
In plants.
In plants only, the cell wall will be there. Now this cell wall, basically, if you remember, it was observed by the Robert Hooke. So cell wall was first seen in cork cells by Robert Hooke in 1665. So when he discovered the cell, during that time itself, he has actually found the cell wall. Now, what happened? It is present. It is present in plant cells. In plant cells, they are also present in the bacteria. Bacteria will also have the cell wall, and also they are present in the fungus. So fungus also will have the cell wall. The only difference is in the plants, the cell wall will be made up of the cellulose, but in the fungus, there is another substance what they are made up of, which is called as a chitin. Yes. Now talking about the structure of it, the cell wall will be basically it is thick. It will be the non-living. At the same time, it is now the non-living, so it means it will be rigid in structure, and also they are permeable. What do you understand by permeable?
Permeable. Semi. I
Permeable
don't remember.
means permeable means it allows the substances to move inside or outside through them.
Acha.
Okay, so they are permeable. They have the permeable covering, which is made up of the cellulose. So now with this covering, what happened? Whatsoever they need, suppose they need the carbon dioxide gas, they can take it easily. They have to release the oxygen gas, they can do that. So that is what it is called as the permeable. Now this cellulose, what they are made up of, it is basically a kind of carbohydrate molecule. Carbohydrate molecule they will be. Now, this cellulose which is present in them, it provides the strength to the plant. It provides structural strength to the plant. So, plants will be getting the strength due to this cellulose, and this cellulose will be present in the cell wall part of it. Okay.
Yeah.
Whereas if plant in the plant, it is made up of in cell wall. As if I talk about the fungus, they will have the chitin where they are being made up of. So that is the difference between the both of them. Clear?
Yes, ma'am.
Now, when we talk about the more detail or depth manner of the cell wall, what happens? The young growing cells, they will be basically. Do you know what are meristematic cells? Any idea?
Merismatic.
I'll write the spelling of it.
I
Mer-
don't, ma'am.
meristematic. I think in your grade 9 only you will be learning. Meristematic means they are the cells which are dividing cells, which can divide actually. So they are present only in the plants. So in the plants, they will have two types of the cells. One is the meristematic, and second one, they are the permanent. So every time all the cells cannot divide, right? Sometime now, once they have divided, after some time, they have to stop dividing. Then only, they can become dead, they can become hard. So when they divide, they are called as a meristematic, and when they stop the division, then they are called as the permanent. Am I clear, Advika?
Ma'am, but then merismatic means dividing cells, right?
Correct.
So when we get a cut on our hand, so to repair it.
This is present only in the plant cells, not in the animal cells.
Oh, okay, fine.
Okay, we are talking only about the plants because cell wall is present only in the plants, not in the animals, right?
Huh.
Got it?
Yes, ma'am.
Okay. So that is what they will have the mesmatic cells through which they will be doing the photosynthesis. They will be helping in storing of the cells. They will be helping, to make the further cellulose through it. That is what they will be doing. Now,
Yes.
the next one is the plasma membrane. What is the other name of plasma membrane?
Cell membrane.
Correct. Okay, both are same. So, plasma membrane and cell membrane, they are same. If we say plasma membrane or cell membrane, we should not get confused. They are same. Now, be it the plant cell, be it the animal cell, both of them will have these plasma membrane. The only difference is that in the plant, the outermost layer will be the cell wall, and then next to the cell wall will be the plasma membrane. But in the animal cell, the outermost layer is the plasma membrane itself. So we can say that the every cell is bound by a thin. So they will have basically a kind of delicate membrane. So this membrane, which is a plasma membrane, that will be delicate in nature and are called as cell membrane, or they are called as plasma membrane. Or there is also one more word which is called as plasma lemma. So when you go for your NEET examination, all they will not be writing their plasma membrane, cell membrane. They will be mentioning the name called as a plasma lemma. Talking about the structure of it, it is basically a transparent. They will be transparent in nature. If we want to see them, then we will be requiring the electron microscope. They are elastic as well. They have the regenerative membrane. What do you understand by regenerative, Advika?
They gen they generate again.
Correct. If something happens to them, then again they will be forming that layer. So they are the regenerative membrane which is present in both eukaryotic and prokaryotic organisms or the cells. Clear?
Yes, ma'am.
And they are present outside the protoplasm. Now, what is protoplasm? Any idea?
Protoplasm.
No, I
I don't
can.
know.
Okay. So when we talk about the protoplasm is C plus N. C stands for the cytoplasm and N stands for the nucleus. So here they are saying they are present outside the protoplasm, which means they are basically present in the space, or we can say that they are present outside the nucleus and outside. Suppose this is cell here, okay? This is a cell membrane here. The center part, it is called as a nucleus, and surrounding the nucleus will be the cytoplasm. So you can see with the diagram itself, the nucleus is there in the center, near to the nucleus cytoplasm, and outside to that is what is the plasma membrane. So that is how they are present outside. Okay, so out of all these things, the protoplasm basically they are the living component of the cells, which includes the cytoplasm and nucleus. So when we talk about both the components, cytoplasm and nucleus together, we say them as a protoplasm.
Okay.
Okay. Now here there is one word like earlier I told you about that, permeability. Permeability, it's basically kind of permeability. How much?
Ma'am, I have a doubt.
Huh?
A little up, can you do? Can you scroll it a little? Over there, P is the protoplasm only, right? Or the
Correct.
plasma membrane.
Protoplasm.
Okay.
The P
Yes.
is here protoplasm. Protoplasm is equal to C plus N. Now, kind of permeability in this, there are basically four types of it. The first one, impermeable. The second one is called as the permeable. The third one is called as semi-permeable. And the last one is called as selectively permeable. So impermeable, permeable, semi-permeable, and selectively permeable. So one by one, I'll tell you what are those. When we talk about the impermeable, it means if a membrane does not allow, just a minute, Dovika. So, um, impermeable means if a membrane does not allow both solvent and solute. Now, do you have any idea what do you mean by solvent and solute?
Yeah.
What they are?
Solvent plus solute equals to solution.
Very good. But what are solvent and what are solutes?
Acha, acha. Those are the components of a mix. See, if we mix solute and solvent, okay, they form a
Hmm.
mixture, okay.
Correct, correct. Huh?
I can explain you in simple words like that only.
Okay. I'll tell you. In a simple example, I'll tell you. Suppose I'm taking the water, okay, and in the water, if I mix something, that is what the solute. Now, water is a solvent in which I am going to mix anything, and that component which is get mixed, that is called as solute. Suppose now I have the water. Now in the water, I want to mix the sugar. So sugar is a solute, whereas in which component I'm going to dissolve it, that is a solvent. So water is a solvent, whereas the sugar is a solute out of it. So solvent, they are basically the substance that dissolve the another substance, and solute are the substances which get dissolved in it. Clear?
Yeah.
Now, in this impermeable, if a membrane does not allow both solvent and solute molecules, so neither they will allow the solvent, neither they will allow the solute molecules to pass through it. They are called as impermeable. Second one, in the permeable, what happened? If a membrane allows both solvent as well as the solute molecules to pass through it freely, then we say them as the permeable. Clear? So in this condition, both will be passed easily. Shall we go to the next one if it is clear?
Yes, ma'am.
Now the next one, which is semi-permeable and selectively, I'll just write them here. Semi-permeable and selectively permeable. Now, in this case, semi-permeable, when we can say them. In this case, if a membrane is there and it allows penetration, it allows only the penetration of solvent molecules. So through them, only the solvent molecules can go, but the solute particles cannot. It does not allow the solute particles, which means I can say that solvents in most of the case will be what? They will be the water. Like for example, if you have the egg, inside when you boil the egg, what is happening? The water goes inside it, correct?
Yes.
Now, automatically, when water goes inside, water is what? The solvent. When the water goes inside the egg, that is what is the semi-permeable membrane. It allows only the solvent to go inside or penetrate inside. Whereas in a selectively membrane, if a membrane allows penetration of solvent freely, but selects the passage of specific solute. So solvent will be allowed as usual. They can go anywhere, any point of time. But what happened? They will select the solute, whether it has to go, it has not to go and all. So the passage of that specific solute particle will be selected. That is why it is called as selectively permeable membrane. Like example, plasma membrane. Plasma membrane is basically a selectively permeable membrane because through this membrane only selected particles, solute particles can go inside or can come outside. Am I clear, Advika, with this?
Yes, ma'am.
So four types are there. Impermeable, it does not allow both solvent and solute molecules to pass through. Permeable, it allows both of them to pass through. Semi-permeable, it allows only the solvent to pass through. And selectively permeable, they allow the solvent to pass through, but at the same time, they will allow only the selected solute to pass through them. Clear?
Okay. Yes, ma'am.
Okay. Now, Where's my pen? No. Here, there's one model actually, which is called as the fluid model. I'll write the name of it. It is called as fluid mosaic model. Or it is also called as sandwich model. Like the sandwich will have, the upper bread and lower bread. In the center there will be a stuffing of any kind of the substances. It can be the potatoes, it can be the onions or cucumbers and all. So that is why it is also called as the sandwich model. Now I'll just draw the picture of it. It will be like this. Outside there will be the circles like this, and they will have the tail coming out of. These are called as the tails. The head and the down which I am drawing, they are called as the tail part of it. Same way, the downside also they will be having the head, and inside they will have the tail like this going to the inner part of it.
This doesn't look like sandwich.
Yes. So, like this, they will have the structure. Now, if I talk about the labeling of them, this head, the circle part, is basically the hydrophilic. PHIL. Sorry, I'll write it again only. HY hydrophilic. It is called as hydrophilic head. Whereas the down one is called as hydrophobic tail. What is phobia?
They will move away from the water.
Correct. That is why they are present inside actually. So hydrophilic, they like the water. That is why the head is outside. Whereas in the case of hydrophilic, they do not like the water. That is why the tail is present inside to them. Okay.
Yeah.
And also outside they have the protein which is called as the intrinsic protein. This intrinsic proteins, what do? They help in passing of the substances. So whatever the hydrophilic head is giving to the hydrophobic tail, then they will be giving to the next part of it. Like this, it will be passing, and ultimately it will be reaching to the intrinsic protein. And now this intrinsic protein will be giving it to the next part of it. So like this, the communication, it's acting as like a communication channel here. Same way, here also in the center, this is called as the intrinsic proteins. So directly the substance will come out here, and then they can go to the next channel of it. Clear?
Yes, ma'am.
So in the year in 1972, basically two scientists, Singer and Nicholson, they were those two scientists who actually suggested a model. It is not off, it should be for model for plasma membrane, and they call them as the fluid mosaic model. Now, according to these two scientists, what happened? They says that according to them, they says that the plasma membrane is made up of plasma membrane is made up of a bilayer of phospholipid. Now, why are they saying bilayers? Can you tell me, Advika?
For the transportation and all.
No, bilayer means two layers they have. This is the first layer, this is second layer. That is why the bilayer. Okay. So they have since two layers. Like in the sandwiches, if we have to make, there will be two layers, right? The upper one and the down one.
Yeah,
Same
but
way.
then why do they have it?
Sorry?
Why do they have it?
You mean why they have the two layers?
Yeah.
For the passage. If they have only single layer, imagine if they have only the head and only one tail is present, then what happened? As I told you, the tail part does not like the water, and if only one layer is there, they will not be able to pass the substances. That is why in the opposite direction, then the other layer has joined there. Got it?
Yeah.
So that is why they're called as a bilayer of the phospholipid, since they have the two layers of the phospholipids, as well as as well as here, they will have the two types of the protein, intrinsic, which is present inside. Two types of proteins will be there. The intrinsic proteins and the extrinsic proteins. So these are the two types of them. Now here, intrinsic means which completely uh, which completely span the molecule floated about in the fluid of the phospholipid. Which means, in a simple language, whatsoever the molecules they are floating, they will hold them and they will take them inside, which means floated about. Whereas in the intrinsic, it is basically which occurs outside, which occur on the outer surface of the lipid protein. So one intrinsic one, where what they are doing, they are taking it inside and then they are giving it to the intrinsic protein, and this intrinsic protein will be passing it to the other channel of it. So like this, both the proteins, they are working together for the movement of the molecule. So they have the two layers for the movement. If they have only single layer, they will not be allowing the water to pass. As the tail part, they have a fear of the water. They do not like the water. They move against the water. Clear?
Yeah.
Now, the functions of plasma membrane. When we talk about the function of the plasma membrane, basically it regulates the exchange of materials between the cytoplasm and the extracellular fluid. So whatsoever the extracellular fluid will be there, they will help them to pass those components in and out. Extracellular fluid is also called as EC. Then they also allows the selected molecules to pass through. So it allows movement of selected quantities of selected material. So they will be deciding how much of the quantity has to come in or it has to go across it. Then it is basically flexible in nature and it is flexible enough to engulf. So basically, they can engulf the large molecule. So whatsoever the large molecule come, they will. So large molecules means here the protein molecules. Okay. So whatsoever the large molecules of the protein comes, they will engulf them. And once they engulf them, they can use them for the further processing it. And at last, they also protects the cell from the injury. Understood, Advika?
Yes, ma'am.
So any doubt in this?
No, ma'am.
So that's about the plasma membrane. A small part of it we have discussed, but still the other things are there. Okay, we'll go to the next one now, which is the diffusion. Any idea what is diffusion is?
Diffusion and osmosis, right?
Yes.
From high concentration to low concentration. Coming
How it
from,
will be happening.
I don't know that.
Okay. Okay. So, do you have the DO at your home?
Dio.
Yeah.
Yes, I have.
Go and get it.
I have Park Avenue.
That's okay. Anything is okay. You can turn on the video so that I can see.
Hmm? Just a second. Where should I be?
What happened?
Okay, fine, yeah.
Ha.
I'll,
So, where is your DO? Park Avenue.
I'll
Okay.
keep it.
So, now what you need to do is you just go in the one corner of your room, spray it a bit, and come back.
I'm not gonna spray in this room, okay? My mom will scold me. Some
Go
other
in
room.
a whichever room you wanted to go and spray, just spray it and come back to your place. But you have to be there for some time, okay, in that room, just in one corner of the room, okay, not in the center, in one corner.
Yes. Okay.
Okay, now be there for 2 minutes and tell me what happened. Something happened? Can you actually feel the fragrance of that Park Avenue?
No. Yeah.
Why so? You have sprayed at the
Because
one corner, right? But you are
yeah,
able to actually get that fragrance at the another side of the room. Why so?
because the molecules in the air they are moving.
Exactly. See what happened when we talk about the diffusion, it is basically the movement of the molecule from the higher concentration to the lower concentration. Correct. Now, what you
Yeah.
did, you have sprayed that in one corner. So, that corner where you sprayed that perfume or the DO has got the higher concentration. Now, immediately when they have got the higher concentration, that particular place, what happened? Those molecules from that higher concentration of one corner of that room started moving in the other corners of it to have
Yeah.
a kind of the constant medium. So until unless from one higher concentration or one corner, those molecules entirely does not spread, they will keep on moving. And after some certain time, you feel that Dio, what you have spread at one corner, the fragrance of it is coming all through the room. That is what the diffusion. Until and unless a kind of the constant condition comes, the molecules will be keep on spreading, and that is what the diffusion. Diffusion does not require any kind of the membrane. Without membrane, they work like you have done with the deo. Another thing you might have seen, right? Your mother might be doing the puja. See, she will be using the incense stick, correct? She will be burning that incense stick in the temple, in the mandir, in the, like, in the small corner of the room. But the fragrance of it will be coming all around.
Yeah.
That is also what the diffusion is.
I've seen that only once or twice in this year, because the whole time I'll be there in my school or else I'll be sleeping.
So that is also what the diffusion is, where the molecules, until unless they do not reach to a constant kind of the condition, they will keep on moving. Then one more example, like if you take the beaker, and in the beaker, at one corner of the beaker, you can just, put the drops of the, might be, the blue ink or the red ink at one corner of that beaker. But after some time, what you will notice that those molecules, those ink, what you have kept on one corner of the water of the beaker, that will start spreading. And after few minutes, the entire water will have the same color. It is not like one side will have the dark color, the other side will have the light color. Correct? The every part of it will have the same color of it. That is also the diffusion.
Yeah.
Okay. So diffusion is a process in which the molecules will be, or substances will be moving from the region of the higher concentration to the region of the lower concentration. And they will be moving until unless they are not evenly distributed through the space wherever they are available. If they are done in the room, they will be moving throughout the room. If they are done in the beaker, they will be moving through the beaker. So whatsoever the space we are using in that entire region, they have to do the movement.
Yeah. Ma'am, I have one question.
Hmm.
Like, not related to the topic.
Hmm.
If we add lemon to baking soda, some reaction will happen, right?
Lemon to baking soda. Vinegar also you can add to. There is a sound coming out. The gas will be releasing.
Ah, okay, that's what I want.
Huh? That gas will be releasing and the hissing sound will be coming like this.
Okay, done.
So, and then if you, when you go to the market, right, there will be the people who will be selling the bouquets and all the flower bouquets. There also the fragrance come. So that is also because of this, the diffusion process of it. So that is how the diffusion will be working. Clear?
Yeah.
So we will be writing the diffusion, might be, some again, then now we will be meeting at 150.
150.
50.
1550.
150. Okay.
Yeah.
I might be 1 2 minute late, but I'll try to start by 150 itself.
Okay.
So somewhere I think 40 minutes class again I'll take it, okay? 40 or 45 minutes whatsoever. But have you understood till here?
Yes, ma'am.
So in the next class we will be completing the diffusion, significance of diffusion, and then we will be starting with the topic called as the osmosis and the types of osmosis solution.
Yeah.
So that will be useful, and some questions.
Ma'am, can you share the notes? I'll write it down.
Let me try to share. Just a minute. Mm-mm. Just give me a minute, Atvika. Let me see how to be. Okay, now I'll share that. Okay, I've shared it, Ritika. Okay, so we'll meet again at 1:50.
Yeah.
Okay, Advika then. Thank you.
Thank you, ma'am. Bye.
Thank you. Bye-bye.